Hello and welcome back. We're going to be diving into chapter 10. But before we get into some of the specifics about meiosis, which is the subject of chapter 10, we're going to take the time to go through some important vocabulary that will save length on future videos. So the first thing I want to talk about is chromosome numbers. So cells, whether the organisms are multicellular or unicellular, can have different numbers of chromosomes depending on the stage that the cell is in the life cycle of the organism. So for cells that are diploid, di, that prefix di means two, and so diploid cells have two copies of every chromosome. Okay. Now, if you remember way back when we talked about our chromosome anatomy lesson, we talked about how chromosomes are linear pieces of DNA. At least in eukaryotic cells, they're linear. In prokaryotic cells, chromosomes are circular. But in eukaryotic cells, chromosomes are linear. And they've got this skinny part in the middle called the centromere. A really important axiom, if you will, is that you count chromosomes by counting centromeres. Oops. The reason for this is going to become clear in a little bit, okay? But just you count chromosomes by counting centromeres. So if there's one centromere, there's one chromosome, okay? So diploid cells have two copies of every chromosome, whereas haploid cells only have one copy of every chromosome. Now, for animals, the only haploid cells that we have are our gametes. And gametes are sex cells, okay? The rest of our cells, the rest of all animal cells are diploid. But that's not true for every organism. So within multicellular organisms that reproduce sexually, we can divide cells into a couple of different categories. Okay, one category is called somatic cells. That's an A. Somatic cells are the body cells. Nothing about somatic cells is heritable. It is not passed on to future generations. So, for example, if you are someone who likes to spend a lot of time in the sun, but maybe you're not real rigorous about wearing sunscreen, and you develop melanoma, skin cancer, maybe you do that before you have kids and you're worried, oh my goodness, I'm going to pass on the skin cancer to my kids. You're not, because those mutations developed in your skin cells. Your skin cells are body cells. Any mutations that you acquired as a result of spending time in the sun without sunscreen are unique to you. They're not going to be passed on to your kids. Somatic cells divide only by mitosis. So as a result of that, other than, changes in the DNA as a result of the copying process or environmental factors, there's no genetic variation from one generation of somatic cells to the next. And when I say one generation, I'm not talking about you and your kids. I'm talking about when one skin cell divides to make 2 new skin cells, that kind of a generation. Okay? Now, then we have germline cells. Germline cells are the cells that are set aside early in development that contain the DNA that will ultimately be passed on through reproduction. Okay? Now, in animals, germline cells are diploid. Somatic cells are diploid in animals. Germline cells have 2 possibilities when it comes to cell division. They can divide by mitosis to create more germline cells, or they can divide by meiosis to create gametes. Gametes are sex cells. So eggs in females, sperm in males. Gametes are haploid. Okay, so again, the germline cells are the cells that contain the genetic information that's going to be passed on to offspring. Those germline cells can divide by mitosis to create more germline cells within an organism, or they can divide by meiosis to create gametes. So different organisms have different methods of reproduction. And again, you've probably learned about this since you were in elementary school. Asexual reproduction and sexual reproduction. All right. Only for organisms that reproduce sexually are we talking about meiosis and germline cells and gametes. Sexual reproduction is also a method of increasing genetic variability within populations. And why is that important? It's important, if you remember, because only populations that have genetic variability have the ability to evolve. So remember, populations are the smallest units of evolution. And in order for selection pressures to act on populations, there has to be genetic variability. Sexual reproduction is one method of increasing genetic variability within populations. All right, organisms that reproduce asexually are not increasing genetic variability. And asexual reproduction is essentially mitosis, where, and this is true especially in single-celled organisms like prokaryotes and yeast or amoebas. There's only one parent, and that parent cell divides by mitosis, which essentially is just copying the DNA and then separating that DNA equally into the daughter cells. So organisms that reproduce asexually are essentially clones of the parent. Only mutations that arise through DNA replication or through environmental factors are going to increase genetic variability in these populations. Okay, so we've talked about chromosome number, we've talked about we've talked about cell types, we've talked about methods of reproduction. So now let's talk about chromosome types. For organisms that reproduce sexually, we can divide the chromosomes into two types: autosomes and sex chromosomes. So the autosomes are essentially the chromosomes that do not determine sex. So the autosomes have nothing to do with whether or not an organism is going to be male or female. The sex chromosomes do that. However, the sex chromosomes have more genetic information on them than just determining sex. So when we get to the end of chapter 11, we're going to talk about sex-linked traits. And so there's a lot of genetic conditions that result from genetic information that's carried on the sex chromosomes that have nothing to do with determining sex, but those genetic conditions are more prevalent in one sex or the other because they are carried on the sex chromosomes. So, for example, human beings, our diploid number, which is characterized by 2N, so remember diploid is when we have 2 copies of every chromosome, our diploid number is 46. Our haploid number, which is 1 copy of every chromosome and is characterized just by N, notice N is half of 2N, our haploid number is 23. Well, in our diploid cells, we have 44 autosomes and 2 sex chromosomes. Different organisms have different numbers of chromosomes, but organisms that reproduce sexually are only going to have 2 sex chromosomes. The remainder of their chromosomes are going to be autosomes. When germline cells reproduce by meiosis to form gametes, in these haploid cells, there's going to be 22 autosomes and 1 sex chromosome. There's a genetic process that allows geneticists to view chromosomes, and this is called a karyotype. DNA is taken from cells that are actively dividing, and notice that the DNA that we're looking at is in the metaphase chromosomes. Okay, now if we look at this, these chromosomes, these drawings of the chromosomes, you'll notice that these are the chromosomes are condensed, right? That only happens in prophase and after. And the chromosomes are pairs of chromatids, okay, of sister chromatids. So after S phase, when the DNA is replicated, each chromosome is a pair of sister chromatids. And the chromatids are held together at a common centromere. And so remember our axiom earlier, we count chromosomes by counting centromeres, okay? So even though there are 2 chromatids here that will eventually become their own chromosomes once they separate during anaphase, right now they are pairs of chromatids. They're held together at a common centromere, so they are considered one chromosome. All right, so what a karyotype does is it takes pairs or homologous chromosomes and it lines them up. So both of chromosome number 1s, okay, both of chromosome number 2s. And karyotypes allow geneticists to do a couple of things. One of the easiest things is sex determination. Okay, so if this is a human embryo or human fetus, for example, you do a karyotype and you can do sex determination. So the X chromosome is much larger than the Y chromosome. The other thing karyotypes allow geneticists to do is to assess for chromosomal abnormalities that may lead to genetic conditions. So, for example, a fairly common chromosomal abnormality would be an extra copy of chromosome 21. Okay, that's known as trisomy 21 or Down syndrome. Okay, karyotypes allow geneticists to see if different chromosomes may have missing parts, if there are missing chromosomes altogether, extra chromosomes. And then these colors are when the chromosomes are stained with different compounds that allow the geneticist to view specific things about them. Okay. Now, could you look at a karyotype and know if someone is going to have sickle cell anemia? No, because remember, sickle cell anemia results from a single nucleotide change in the hemoglobin gene. You cannot assess individual genes with a karyotype. Karyotypes are specifically looking at chromosome number, chromosome structure, So bigger things, you can't identify specific gene defects looking at a karyotype.